All right, welcome back. We're to the last section of Chapter 7, and we're going to talk a little bit about bulk transport and cotransport, which are two unique ways of moving materials in and out of the cell through the cell membrane. Cotransport is happening when the active transport of a solute indirectly drives the transport of other solutes. An analogy of this would be, for example, pumping water up a hill so that it can do some work, moving turbines or whatever, as it comes back down. And a common example would be that plants are going to be using hydrogen ions that are generated by their proton pump to drive the active transport of certain nutrients into the cell, certain sugars. They would not be able to get through the cell membrane without the addition of the hydrogen ion. So the hydrogen ion is just being pumped out as it is against the concentration gradient. So they're going to come back in. It's going from low concentration to high concentration, so it's naturally going to move back in. And as it's moving back in, it's going to take with it some sugar molecules. In this case, with the picture that you see on the screen here, it's grabbing sucrose molecules and facilitating the delivery of sucrose into the plant cell. Again, so this indirectly is being involved in the movement of sucrose into the cell through the proton pump. And there are a number of examples like that where hydrogen ions are being moved, or potassium ions are being moved, or sodium ions are being moved. I guess a good example of that would be the example that the book gives you about diarrhea. This is a big problem in third world countries. It's not really a laughing matter because a lot of people, because of poor sanitary conditions, suffer from diarrhea because they're drinking water that's contaminated. Well, the problem is food materials are moving through your digestive system. It's doing so at a rather slow pace. It takes about 12 hours to digest a meal, which gives the intestines plenty of time to pull the water out, to pull the ions out like potassium, etc. Well, if you have diarrhea, it's not taking its time moving its way through the digestive system. It's moving very rapidly through the digestive system. So you're losing water, you don't get to reabsorb that water, and you're losing some really important ions, which can cause death in a lot of individuals. So that would be an example of cotransport. Medicine has developed a number of different drinks that are resupplying not only water but the ions, kind of like Gatorade, to the body, and that will then hopefully replenish the body until whatever pathogen is causing the diarrhea gets taken care of. All right, so bulk transport, moving big stuff through exocytosis, endocytosis across the cell membrane layer. If it's a small molecule, it can pass through channels. It might even be able to move right through the lipid bilayer. And so there's not going to be as much work involved as if you have some really huge polysaccharides or a bunch of big bulky proteins that are in desperate need of being either moved into the cell or being moved out of the cell. So this definitely is going to require an input of energy. It definitely is active transport, but even on a larger scale than what we talked about in class. So here are a couple examples. Exocytosis, you know that the word exo means out. So we're going to take a vesicle, a transport vesicle that is full of something that needs to be released outside of the cell, and we're going to fuse it to the cellular membrane. In the process of doing that, the cellular membrane is going to open up, and all this stuff will just be dumped to the exterior. Here's an animation that hopefully you'll be able to hear okay.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
In endocytosis, the cell takes in huge molecules, and it's going to be the reverse of exocytosis. So we're talking about a pretty similar concept. We're going to have these large molecules that are going to be engulfed by the movement of the cell membrane around that material, kind of creating a vesicle or a vacuole. And there are three different types of endocytosis. We have phagocytosis, which is basically what you'd find in white blood cells. Pinocytosis would be the movement of liquids in, and receptor-mediated endocytosis, which we'll talk about in a little bit. So cellular eating, cellular drinking, and receptor-mediated. Here is the animation for exo and endo.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
All right, and we saw, an example in the last animation where phagocytosis is at play. We have some very large material that we want to engulf and bring into the cell. And so if you're a white blood cell, let's say you come across a bacterial molecule, or not molecule, bacterial cell in the body that you want to get rid of, you're going to create a vacuole around it, and then you're going to bring a bunch of lysosomes to that vacuole, dumping their enzymes into it, thus breaking that material down. Here's pinocytosis.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
In receptor-mediated endocytosis, binding of ligands to receptor sites triggers a vesicle formation. So in other words, here we need some assistance. We need to tell the cell, hey, there's something out here that you need to bring in. And so there'll be a receptor site on the external surface of the membrane, and a ligand will come attached to that. That'll send a signal to the cell, hey, we got to move the cell membrane, create this vesicle, and bring this material in. And this will demonstrate that here.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein-cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
All right, and that is a very specific thing for certain molecules that need to be brought into the cell, and they're going to be tied directly to the activation of a receptor on the surface of the cell membrane, like you see here. So these receptor-mediated ones, if they're not triggered by a ligand, if that ligand doesn't attach to the receptor, it will not bring this material in. So pinocytosis, again in review, is bringing in droplets of liquid from the external cellular material. Phagocytosis is using the cell membrane to wrap itself around a food particle or bacteria, whatever, to create a temporary vacuole that, again, we're going to bring lysosomes to. The lysosomes are going to dump their enzymes inside and start breaking this molecule down. That ends chapter 7, and good luck on the exam.